Hello and good afternoon, friends. Welcome to the CEC-UGC live lecture. Dear friends, as you know that we carry different subjects, that is we talk on different subjects, and under these subjects we talk on different topics. Sometimes we carry a particular series for you, and I would like to tell you all that you know that since our previous two sessions we have been talking on non-Mendelian genetics. Dear friends, today also we are going to talk on this particular series, and we could say that this is the third lecture in this series, and today in the lecture itself we are going to talk on quantitative or multifactorial inheritance. Dear friends, in a previous session our expert asked you certain questions. I hope that you might be knowing the answers of those questions. If not, no need to worry, because our expert is here with us and he is going to answer all the questions what he asked in the previous session. He is going to recall what we did in our previous session. So without wasting time, I would like to invite once again Dr. Johansson. He is an Associate Professor in Department of Botany, Deshbandhu College, University of Delhi. So Dr. Johansson, welcome once again to the CEC-UGC live lecture.
Thank you so much. In our last lecture, we summarized the aspects of penetrance and expressivity. These are essentially non-Mendelian, and they were summarizing in a sense that mere presence of a gene would not always ensure that the phenotypic expression is the one that we expect. For example, if a gene is completely penetrant, then all the individuals will show the same type of expression. If it is not, that means there will be a varying degree of expression even though the genotypes are the same. Now, this landed us in a lot of trouble as far as making of pedigree charts was concerned. And we also summarized that the genetic constitution of rest of the genome is also important, because it is going to also influence the overall expression of a particular gene. And last but not the least, it is not the genes alone. They have to, at one point or the other, interact with the external environment. So, we ended on a note whether it is nature versus nurture, or is it nature and nurture. I would just conclude that it is both. Many a times one of them and the other times the other. Today, we begin with what we ended, like these some innocent questions that we asked, and perhaps you are ready with the answers. If not, I would just try to recapitulate, because these are the questions which concern all of us all the time. And if we have no knowledge of genetics and to say science, we lend ourselves into a lot of trouble, and we come into the grasp of various blind faiths and also misconceptions. To begin with, do you think that if parents are small or are of medium height, is it necessary that all the children of this couple would also be dwarfs? They will never be tall? You see in your own family, do all your brothers and sisters have the same skin color as the parents? There are people who are fairer, there are people who are darker. And coming to intelligence, we have a whole list of Nobel laureates in different disciplines of science and technology and humanities. Is it necessary that they have equally intelligent parents? With due apology to the parents, is it necessary that all the intelligent parents always have equally intelligent children? The answer to all of these we are going to attempt in our lecture on quantitative genetics. And if you are given a mathematical problem to solve to the entire family, is it necessary that all the members can solve this problem with equal finesse? Perhaps not. And a very common aspect of eye to eye contact is Do all in the family have the same eye color? Perhaps never. Some more questions we can ask ourselves. You go to a field. Do you find that all the plants in a field are either tall or short? Are there not any intermediate height plants? So, can we just distinguish this plant segment into tall and dwarf? Perhaps never. And why do we say that my brother is above average? My father is below average in terms of height. My mother is short, but I am above average. So, that means why are we taking averages if we are sincerely following Mendelian principles, because it only says either tall or short. So, that means why is it there is so much of variation in size, shape and color of fruits when you go to an orchard. A very simple observation would be, when you go to a hospital, why are different babies born with different weights? Why cannot they be classified as a heavy baby and a light baby? You have above average, you have a bony baby, you have a skinny baby, and you have a very, very small mouse-like baby. Why do we categorize them with such adjectives? So, the time has come to think that in Mendelian genetics, we have always been talking about the alternative forms of a gene being two. So, we always say either it is this or that. That means we have only two alternatives. But then today what we are going to discuss is, in addition to this and that, we also have that too, which means we are dealing with more than two alternatives. If we just glance at a cluster of tomatoes, we find that there is a whole lot of variation in terms of size, in terms of color, in terms of shape. Although the number of chromosomes is the same, which is 12, then why is it that we have such a huge variation, and the statistical methods, it would be difficult to find which parent is going to give rise to which parent. Even their internal fabric is also highly variable. So, that means, can we explain these phenomenon on the basis of just Mendelian principles of short and tall or large or small? Perhaps never, because we find a whole spectrum of intermediate forms. How do we account for that? If we look at another flower of the quantitative variations, it is Gyladia, and we find that this particular flower has all different types of diameters. The number of floral parts in terms of calyx and corolla are highly variable, and so are the shades of the petal. It is the same variety of flowers, but then why is it that we have such a huge variation? We can also find this in the bract color of another plant, which is the Indian paintbrush, that is botanically Castelligia hispida. There are extreme ranges of color varying from light yellow to bright red. But if we see the phenotypic range, it is showing us different hues of orange, yellow, up to flaming red. Now, typical Mendelian genetics cannot account for these intermediate forms. We have to think of something else. A very common observation in the market is the paprika fruits of different shades. We are accustomed to just green, but now we have different shades of mauve, purple, orange, or even red and yellow, of course. So, that means, how do these different shades come along? Genetic engineering, gene insertion comes much later, but then in nature also, all by themselves, such type of variations do occur. We should first discuss, how did this whole idea of polygenic inheritance actually begin? The history of polygenic inheritance dates back to the year 1760, when the famous biologist called Reuter, he crossed tall and dwarf varieties of tobacco, that is Nicotiana tabacum. To his surprise, he found that the F1 progeny were not all tall, there were some intermediate height plants also. Now, today you could say that it is a case of incomplete dominance. He could never understand what was going on, and to add to his surprises, the F2 progeny showed a continuous gradation from dwarf plants to tall plants. Now, this is not what we expect out of a Mendelian cross, and by the way, Mendelism was not there, because this work of 1760 was a bit earlier, and therefore, since the Mendelian principle was not there, he could not explain how did he get intermediate forms and how to account for a continuous gradation, because we would have thought either tall or dwarf. Sir Francis Galton, who was the first cousin of the legendary Charles Darwin, in the later half of the 19th century, along with another scientist Carl Pearson, they were the first to recognize certain metric traits. Now, what are these metrical or metric traits? They are traits like human height and human intelligence. Galton was a very serious proponent of intelligence, and the intelligent people should have their genes propagated further. We would not go into the controversy of eugenics, but it was very difficult to detect the inheritance patterns of intelligence and human height with the help of routine or conventional Mendelian methods. So, that means if such particular traits were really or actually heritable, how could we detect them? So, therefore, new statistical tools, new statistical techniques were used to find out different types of variance, levels of heritability, the mean values, the variance values, and then it was found that these particular traits were actually quantitative. That means they were not just about tall or short, they were about how tall or how short. That means now people started thinking in terms of quantitative terms, and perhaps these genes could add into one another. So, this was the beginning of quantitative genetics, and by this time two schools of thought really emerged. There was one thought of Mendelians, which was propagated by William Bateson himself and the great Hugo de Vries. They believed that all the characters which are heritable are qualitative and discontinuous, whereas Galton led school of biometricians, they were convinced that the heritable variations were basically continuous and they were quantitative, and they said let us talk in terms of numbers, let us talk in terms of units and not a phenotype of dark and light alone, and they believed that these particular traits could also be heritable. So, that means this controversy reached its zenith or peak, because we believe till then that genes are discrete and they should also lead to discrete phenotypes. That means our mindset till then was that whether it is capital R or small r, it has to be round, and if it is homozygous small r, that means it has got to be wrinkled and there is nothing in between, there is nothing intermediate. Science had to wait till 1918 when Sir Ronald Fisher wrote a revolutionary paper, and this research was basically showing that the factors which are showing inherently discontinuous variation, they will also cause genetic segregation, and they can be later translated into continuous variation. And he for the first time also suggested that these characters were perhaps quantitative too. So, till now we were just talking about qualitative characters. The real work began in the line of quantitative genetics with the work of Wilhelm Johansson. As we all know, Johansson was the person who gave the term genotype and phenotype, and Johansson worked on beans. He was the first person to have given a quantitative approach. He weighed the seeds, and to his surprise he found that the weights of seeds were extremely variable. So, he said, what do we mean when we say a seed is heavy or it is light? We should now ask ourselves a question, how light or how heavy they are. And he saw to his surprise that the weight was ranging from 1 centigram to 90 centigrams. So, this was enormous, and it was also shattering all the laws of statistics. I mean, it could not be statistically insignificant. He bred 19 pure lines, and he observed variation in each of these lines, and his work indicated that perhaps not just a single gene was being operative, it was another gene in addition also. So, in effect, the Mendelians as well as the biometricians, both of them had some truth in what they were believing. So, that means both the schools of thought were perhaps partly correct. Johansson was the person who also tried to separate the role of environment from genetics, because he appreciated the term phenotype and genotype. He was the person to have suggested that the phenotype of an individual would depend on its genotype, no doubt, but then the environment will also have a significant role to modify what is present in genes. So, he performed a set of experiments in his first, and they were very statistically sound. And in the first experiment, when he weighed a group of beans and he also selved them and weighed their progeny, he was surprised that when the weight of parental beans was smaller, their progeny in the next generation were also comparatively lightweight, and he saw an enormous variation in the weight. So, that means he said there is no point classifying the seeds merely as heavy or light. We have to quantify our results. Johansson's second experiment was even more interesting, because now he wanted to find out that does environment also play a significant role. So, when he had 10 generations of inbred lines for beans, he was selfing them all the time. He found that the percentage of heterozygotes became less and less than the original level after each generation. Now, these were pretty expected results. The third experiment, which now forms the foundation of modern artificial selection. Today, we can select different breeds of horses. We can to make them as good race horses. We can select large apples. We can select large sized tomatoes, which are economically more viable and more glamorous, and they have more content. So, the beginnings were made by the experiments, the pioneering experiments of Johansson, and then he worked out the artificial selection. All the random based population he screened, and he found that the results were perhaps a mean of the two parents, and that means if he could take two robust parents, there would be a mean of this robustness in the next generation. So, that means we would have better and better progeny. Needless to say, this procedure is absolutely unworkable if we are talking about inbreed populations, because in inbred populations there is no genetic variations, and if there are no genetic variations to inherit, then how are the new recombinants going to come? So, even if we select the best parents, we go on and on breeding them, no new combinations are going to arise. So, in other words, the main interpretation of Johansson's work was that if heavy parents are taken, they would give rise to heavy offsprings, and the light to the light. So, that means he did not have any doubt on Mendelian genetics. So, that means there was a definite relationship between parent and offspring weights, no doubt. At the same time, there is a variation, and this variation in the offspring weights can be attributed to both the parental characters on one hand as well as environment on the other. So, this signified that the role of environment in genetics in modifying, say for example, such a simple feature like the weight of the seeds. At the same time, if we were talking about average, he talked about a phenomenon which is called as regression to the mean. What he really meant was that most of the offspring taken from the extreme parents, which means which were on the edge of the graph, they were either very light or very heavy. They were having more averages than their parents. So, it means the parents of the two extremes would always enjoy the benefit of the environmental conditions. It is not necessary that such benefits may always be inherited. They may or may not be. Just to illustrate the point, if we have a look at the fact that environment can also play a significant role in manipulating or in modulating the phenotypic variation, we can have the example of a yarrow plant which is Echillea. Now, depending upon the elevation, that is the altitude at which this plant is growing, if a plant population is taken say from 100 feet above sea level, plants have been recorded which are up to 1 meter high. That means it is a case of luxuriant growth. As the elevation increases, as the altitude increases, so say for example to 4,600 feet, the size becomes smaller and smaller. We know, students of ecology know that by a line transit, if we go from a plain to the mountains, of course, a gradation in size is there, because there are so many other factors like wind velocity, like gravity, like availability of water, and of course, sunlight, and not to mention the modulating effect of temperature, they are all operative. And finding the extreme, that is at 10,000 feet above sea level, the plant would be so small, it would be particularly insignificant as compared to say 1 meter or 100 centimeters of length of the same relative. So, that means genetically these plants are the same, but then their morphology is so very variable just because of growing under different environmental conditions. Maybe later we could translate this into a more statistical approach when we attempt to study what happens to the behavior of two identical twins separated at birth and given two different, altogether different environmental conditions. Is the behavior of their intelligence, their personality, their way of life, is it really different? Well, the answer is yes. That is why you see so many movies, they are made on the different behaviors of two different twins who are separated at birth. So, that is fantasy, but then really based on quantitative genetics. It was another scientist by the name Yule who really took up this work of Galton and later of Johansson, and he said for the first time on his work, depending upon his work, that more than one gene was definitely responsible and the variation was quantitative, because somehow he found that a discontinuous characteristic will show only definite phenotypes like tall and dwarf, and therefore, we have only two phenotypes at the end. Whereas, if we have a continuous characteristic which is a quantitative one, we will have so many intermediate phenotypes: tall, slightly taller, dwarf, slightly smaller, and various intermediate types. And if we were to see the height or the phenotype, it would show a Gaussian or a pyramid-like variation pattern, where there is a continuous gradation of height starting from dwarf to the tall one. So, it is not an abrupt tall or dwarf, it is dwarf, taller, much taller, and even taller, and the tallest, depending upon how many genes are involved. So, that means, to summarize, we have two types of variations. Now, this appreciation is a must before we come to the actual calculation of how many genes are involved, what is the formula which is underlying the quantitative gradations, how many types of phenotypes would be there, and finally, some numericals to prove our point. So, that means the Mendelian traits are basically qualitative, they are revolving around kinds. Kinds means the phenotype, whether it is round, whether it is wrinkled, or whether it is black and white coat color, or the cattle whether they have horns or they are hornless. We know that such type of genetic control is usually by two alleles of a gene. Sometimes there could be multiple alleles also, as we saw in one of our previous lectures of multiple alleles and the blood group genetics. Here we would find that environmental modifications would be minimum. I would not say no, because environment does modify us and as a result distinct phenotypic classes are observed. So, that means such variations are discontinuous. You have black and white, you have round and wrinkled, but then there is no gradation which is visible. On the other hand, in sharp contrast, we have the quantitative traits, and these traits are also called the metrical traits. Later we would have various synonyms for these terms like Galtonian genetics, polyfactor inheritance, multiple factor inheritance, additive inheritance, and many more. So, that means here we are not talking about red and white, we are not talking about tall and short, we are talking about quantitative parameters, which means important, measurable, not just qualitative, measurable phenotypic traits, which means height, how high, weight, how much, shape, pigmentation, and metabolic activity, behavior, eye color, and also very interesting aspect in medicine, susceptibility to pathological diseases. There are many members of the family who are really vulnerable, whereas their kin are not that. Then intelligence in human beings, it also varies in degrees. Number of flowers per branch, for example, number of seeds per fruit, the height of plants. We just observed the experiments of Johansson in seed weight. Even in poultry, we find number of eggs given by the hen, and also variable milk production. And the factor which is so dear and paramount to a farmer, that is the grain yield per acre. That means, why do we have different varieties which can have more grain yield? So, that means there is no clear cut difference between the individuals. We cannot just classify them as tall and dwarf. There is in fact a spectrum of phenotypes. That means this is going to blend in a very imperceptible manner from one type to the other. That is why it would give rise to a Gaussian or a pyramidal type of a graph. In other words, such variations are not abrupt, they are not discontinuous, they are of the continuous type. What would be the genetic basis now for such type of inheritance? Our familiar mindset is that if there is one dominant allele, okay, it should be given an uppercase and all the other alleles would be in the lower case because they are recessive ones. For example, here we take the example of the baby mice. The mouse is recessive for a particular character, its weight is 6 grams. The dominant one, now whether it is homozygous dominant or heterozygous, regardless, it is 14 grams. So, that means this is entirely different to what we are studying today, and that is the additive model. In an additive model, we find that if there are no dominant alleles, the weight is 6 grams, fine. But if there are 2 dominant alleles, then only it is 14 grams. If it is 1 in place of 2, then it remains only 10 grams. So, where did this third phenotype come? This is the effect of a non-Mendelian type of existence, what we call as an additive gene, an extra gene, which we would now call as a polygene. What would you expect if we were to have a dihybrid cross with 2 genes? We would express that the number or the types of active alleles would be 4, 3, 2 and 1. So, at every point we are talking about a quantitative approach. In a normal Mendelian genesis, capital A is equivalent to capital A small a, but here it is not, and as a result we would find that the average phenotypes are different. AA capital is different, it is only represented by 1/4, and capital A small a is different because it is in 1/2, which is 50%, but then its intensity is different. So, that means in effect we have 3 types of phenotypes instead of our normal expectation of a Mendelian cross of 2 phenotypes. So, if we summarize the phenotypic values, we were having now 1:2:1 in terms of 3 phenotypes, which is not heard or not seen in a conventional Mendelian cross. Likewise, we would find that if the genotypes and phenotypes of a dihybrid cross do not have any active allele at all. Now, here what do we mean by an active allele? We mean that there is no dominant allele at all. In that case, the frequency of that particular phenotype will be entirely different. And in a dihybrid, in effect we have 5 types of phenotypes in the ratio of 1:4:6:4:1. This is entirely different from a normal dihybrid cross. Likewise, the ratio we will just derive how did 2n plus 1 come into existence. So, that means according to this particular rule, the number of phenotypic classes would be obtained. The classical experiments of H. Nilsson, Ehle and East on the kernel color of wheat, because we know wheat has different colors, it proved that the genes are additive, and if 2 genes are involved, they would add to the color of the grain. If the number of capital genes is more would be the intensity of the grain. As we can see, If there is one gene pair, then the ratio is 1:2:1, again a pyramid. And if two genes are involved, then we will talk in terms of 5 phenotypes ranging from two extremes, 1, and the maximum number, that is 6 out of 16, would be intermediate. So, the ratio becomes 1:4:6:4:1. 1 out of 16 would be dark red, extreme, and likewise 1 on the other extreme would be white, and there would be intermediate ones. But 6 out of 16 in the beginning, in the middle, would be medium red. So, that means this type of graph is characteristic of quantitative inheritance. So, in other words, we have a bell shaped curve or a Gaussian distribution, and when we are making a graph, we have to be very, very careful whether we are talking about variance, that is the width of this graph, or are we talking about the midpoint. So, that means these are the continuous traits which can be before, on, or after the threshold. This is the basis of determining the future statistical concepts of polygenic inheritance. That is, we have to take the peak of variance or the width, and there are some traits which would be shown only when the threshold is crossed. There are so many pathological conditions in our body which we would have an opportunity to discuss in some of the later chapters, which are governed by the threshold model for polygenic inheritance. Now, how do we calculate the number of polygenes, whether it is 2 genes involved or more? So, that means we know if the ratio of the F2 individuals are resembling either of the two extremes, as I said in 1:4:6:4:1, just 1 out of 16 was one of the extremes. So, for example, if we have 1/4 of n, we want to know the value of n, and that means the extreme phenotypes are 1/4 n, and if we have 16 progeny altogether and 1/16 is red and the other 1/16 which forms the other extreme is white, that means according to the formula of 1/4 n, the n would have a value equivalent to 1/4 n would be equal to 16. So, the value of n would come out to be 2. In other words, n is equal to 2 and number of phenotypic categories which are observed are, according to the equation, 2n plus 1. That would be our basic formula which we can put on all the cases. Give you an example. If 2 polygenes are involved, so n is equal to 2 and we use the formula of 2n plus 1. So, that means the number of phenotypic classes is 5, which we just saw in terms of 1, 4, 6, 4, 1. And in case of a tri-hybrid cross where 3 polygenes are involved, the value of n is 3, which means 2n plus 1, which means 6 plus 1. So, there are 7 phenotypic classes, and these particular phenotypic classes will be now represented by 7 different gradations of intensity. Some would have intensity of 6, 5, 4, 3, 2, 1. This would be very well exemplified by our own skin color. So, that means we have a formula for calculating the number of polygenes, and therefore, if we know the number of phenotypes, we can immediately conclude as to what is the number of polygenes which are involved. For example, if we have 9 phenotypic classes, that means it is the number n equivalent to 4. In other words, it is the 4 polygenes which are involved in this system. So, that means how do we define polygenic inheritance? Number 1, this is a quantitative inheritance. Here the gene loci are adding their intensities or their actions. So, that means the influence on the phenotype is going to be in an additive manner. In other words, more would be the number of dominant genes, more would be the intensity of that particular character, be it in terms of height, be it in terms of skin color, be it in terms of darkness of our eyes or the weight of seeds and so on and so forth. So, that means another point which we have to remember here always is that each of the polygene produces an equal effect. To exemplify this point, the contribution of each additive allele will always be constant. For example, if there are 3 genes which are operative, we find that the value of A is equal to B is equal to C. Now, here we are not bothered which of the allele is present. We are only concerned how many, which means as we can find A, B, C will have the same effect as triple A or for that matter triple B or even triple C or any of them represented twice and one others. So, our emphasis is how many and not which ones, because A is equal to B is equal to C. So, it is always the dominant alleles whose number is important, and if we are talking about their relative intensities, then we find that if we have 3 of these genes any, they would definitely be more intense than any of the 2 put together, and they will be much greater than any one present alone. So, this is pure quantitative expression. On the other hand, there are non-contributing or non-additive alleles which are in the form of the recessive ones. But then, do we say that a person who has all the recessive alleles does not have a height at all? Well, that would be wrong to assume. The right answer here would be that he is on one of the extremes. So, he or she would show the minimum intensity of that particular character. We will clarify this in some of the subsequent numericals. So, that means each of the dominant gene is incompletely dominant over the other gene, and therefore, the expression to conclude is 4 capital genes would be greater than 3 capital genes, any of them would be greater than 2 and would be greater than 1, and the minimum would be shown where all the alleles are recessive. So, that means it is a quantitative approach. The kernel color of wheat we just saw has more of the statistical connotations. Now, we move to the eye color, which is so very frequent. We see so many people around us who those are dark eyed, brown eyed, and people with hazel eyes, blue eyes or even green eyes. Now, this is a character which is based on the different genes. It is also an entirely polygenic trait, and it would also show a characteristic bell shaped graph. That means, if 2 genes are controlling the character, we would have an extreme of dark eye and an extreme of blue eyes, and this would be in the form of a Gaussian or a bell shaped graph, and this would be 1 by 16 would only be blue eyed, the maximum would be 6 by 16, and you can see how wide this graph is when we were talking in the earlier graph, that is the pitch is also important. So, the threshold would be somewhere here, and 1 by 16 only is the other extreme. So, that means if there is a marriage between a dark eyed person and a blue eyed person, regardless of whosoever is a male or female is different, because it is not a sex linked character, the maximum probability of their children having say intermediate color eyes would be 6 by 16, although it is possible that in one of the 16 chances on both the extremes, the children would have the eye color like one of their parents. The slide here exactly exemplifies as to what are the different hues or the colors of the human eyes. They would vary from light blue to dark brown, and it depends on the number of such genes which are present in a population. For example, in India, we have less genes in a population for light eye color. So, that means anybody with a light eye color is taken as an exception. And likewise in Europe, the gene for the cumulative genes for dark eye color are less. Now, this is a question to be discussed in population genetics as to what is the frequency. Another very important case of quantitative inheritance is seen in terms of a new term which has been given, and that is dermatoglyphics, which is the skin writing. Many of us are followers of palmistry. We find that there are ridges and grooves in our fingerprint patterns, and they are different for each individual. It is believed now that these particular ridges and grooves in our fingers and toes, they are also governed by polygenes. Interestingly, we would find that this can be helpful to us in even finding out various conditions of chromosomal abnormalities. This particular slide also shows a typical bell shaped graph, but then if we ask ourselves some interesting facts, and that is a child with Down syndrome, that is trisomy of a chromosome number 21, would show us an irregular ridge pattern in the fingers. We will immediately come to know that there is something wrong with this child. The patterns are not the same. And if we were to compare two identical twins, they are genetically identical. The southern blotting, their DNA fingerprinting would show identical bands, but their fingerprints, actual fingerprints, which are showing the patterns of ridges and grooves, will be different. Why? Because during 6 to 13 weeks of prenatal development, the fetus touches the finger and toe pads to the wall of the amniotic sac, and it is here that the environment changes the ridges and the grooves of the finger patterns. So, can we really conclude that the destiny of each child is clasped in his fists? Well, I would not like to comment yes or no, but I would only as a student of genetics comment that yes, the two identical twins will have two different fingerprints, and of course, their destinies are bound to be different, their behavior has to be different later. The most common observation which we find in ourselves is our skin color. Skin color is known, and these are the classical examples and the research of Davenport, and because he was working on the Negro population in America. We find that 3 genes control our skin color, and it is the intensity of our melanin which is responsible for this. So, any person who has all the 6 recessive alleles would be pure white, and the one with all the 6 dominant ones would be a Negro, and their child would have 3 genes, 3 capital ones. So, it would be a mulatto or brown. For example, it would be wrong to call some of the legends of certain sports to be black, because they are actually not black, they are browns, they are mulattoes. So, we have to really distinguish what skin color are we talking about. And there is a typical graph which is made here for the bell shaped one, which indicates as to we have no dominant allele, 1 dominant allele, 2, 3, 4 and finally all 6. So, we can see the gradation of skin color from white, very fair, fair, mulatto, dark, very dark and finally a negro. So, these are some of the aspects of the quantitative inheritance. We can exemplify our talk with the help of certain numericals if we wish to talk about such points. But then these numericals would require foundations of the type of formulae that we have discussed, and that is 2n plus 1. If we could talk about the human height, the numerical which we usually ask is, which we see around ourselves, a male is 160 centimeters tall and its genetic constitution carries 3 capital genes. He marries a lady with a genotype of 2 capital genes and she is 155 centimeters. A common man would say, okay, both the parents are below average, their child is bound to be shorter. My question is, can their children be taller than the father? If so, what would be the maximum height that their children could achieve? On the other hand, can their children also be shorter than the mother, because mother is only 155 centimeters, and if short, how much shorter? So, that means in terms of quantitative genetics, we have to talk about measurement. So, it is not a case of taking a mean, that is father plus mother by 2. It is a case of quantitative genetics. See, father has 3 capital alleles, mother has 2, and father can contribute in the sperm maximum of only 3 capital ones and mother can only 2. So, if the ovum and sperm having the maximum alleles are fused, let us see what is the contribution of a dominant allele as such. Father with 3 alleles is 160, mother with 2 is 155. So, that means the difference in their capital alleles is 1 and 1 is equal to 5 centimeters. So, that means the contribution of 1 capital allele in height is equal to 5 centimeters. This will help us to calculate what is the base height. Father is 160, he has 3 dominant alleles, that means their contribution is 3 into 5, that is 15. If these 3 were not there, father would have been 145 centimeters, which is his base height. Likewise, the mother has 2, she is 155 and therefore, the contribution in her height of the dominant alleles is 10. So, she would have also been 145. So, 145 is the base height. Now, what could be the maximum additive value of their children? Father can contribute in his sperm maximum of 2 alleles, because he is already 1 capital A and 2 capital B. So, only 1 of the B can come. So, that means father can contribute 2, mother can contribute just 1. In that case, maximum additive alleles in a sperm would be AB and from mother it would be AB. In that case, 145 would be the base height, maximum genes which could come from the parent could be only 4, and in that case 4 means 4 into 5 20 and added to the base height it would be 165. So, the answer to the first question is yes, the child can be taller than the father. How tall? One could be 165. The second question, can there another child be shorter than even the mother, because mother is 155? Yes, because the mother has also another ovum which does not contain any of the dominant genes and father would have the minimum as 1. So that means here the additive alleles would be small a, capital B, small b. So there is only one. So that means we have to add the contribution of just 1 to 145, and that means 145 plus the contribution of 5. A child, regardless of the fact whether it is a daughter or son, because as I said repeatedly, it is not a sex linked character. So a child could even be 150. So the child could even be shorter than the mother. A classical photograph which has been taken many years apart is called as the living histogram. It was taken by the genetic scholars at the University of Connecticut in USA. The first photograph is taken in 1913 perhaps, and it shows a large gradation of different types of individuals. Their maximum height at that time was 5 feet and 9 inches. And in 2010, a similar photograph was taken by the modern faculty, and here the maximum height seems to be 6 feet and 6 inches. Are we really getting taller by the day? Perhaps yes. It is not due to genes alone, it is our environment and nutrition alone also which is contributing. So that means, to summarize, quantitative genetics is an additive genetics of various polygenes together, and also taking into account the blend or the influence of environment. Maybe in one of the subsequent lectures, we will have a more complicated discussion on the aspects of variance, statistical aspects, how the behavior of different twins varies. How is it that we can take a quantitative approach to various diseases as they are being shown by the family members in different concentrations, in different intensities. Thank you.
Definitely. Thank you, sir. Thank you so very much. We would be conducting more and more sessions, and we would be inviting Dr. Chauhan again and again, and it would be our pleasure if he keeps visiting us and exchanging, sharing information with the students.
Thank you. It would be my pleasure.
Definitely, it would be our pleasure too. So dear friends, if you want to access this lecture particularly, then you can watch this lecture on YouTube, as we are going to upload this on YouTube very soon for your convenience, so that you can view this lecture the number of times you wanted. And for your feedback, you can mail us at info.cec@nic.in. We would be meeting again. Till then, take care. Goodbye. Thank you, sir. Thank you so very much.
Thank you so much.